Thanks very much for giving me the opportunity to give you a very quick introduction to the services we have from my team. So I run the Microbiome Informatics team at EMBL-EBI. So I think Alex already alluded to this from my research team that my team runs the MGnify resource, which is a free-to-use resource for the archiving, assembly, analysis, and browsing of microbiome data. Now, that's a bit of a catch-all. I appreciate that. But really what we try and do is we try and collect data sets which are either submitted to one of the INSDC partners, or you can come through us and we will broker it through to the European Nucleotide Archive data sets. And then we will potentially perform analysis depending on the data type, but we'll also perform analysis of assemblies, just the raw reads, or if it's metabarcoding data sets. So we try and cover the full spectrum of microbiome nucleotide sequence data. And I'm not going to go through all of the views, but we provide a veneer of a website onto this data so you can start browsing and accessing that data and you can search it. But over the last few years, metagenomics has become our mainstream analyses, and also the generation of not only assemblies, but trying to recover genomes. So here in this slide is just a growth of metagenomics data over time, particularly assembly data. So along the x-axis is years, along the y-axis is the number of submissions into ENA. And the blue line at the bottom here reflects actually traditional sort of isolate sequencing and assembly. And what you can see is the green line represents metagenomic and metagenomic assembly. The solid line represents the totality, whereas MGnify is the large dashed line here. And so you can see that as we sort of really switch this on into production in 2019, that really we've sort of had a really sort of big increase of assemblies. We actually, that plateau that you see there is because we broke INSDC because there were just too many assemblies, and there are now specific layers in the European Nucleotide Archive for collecting up these metagenome assembled genomes. So this service is free to use, and you can actually request the analysis of your own data or of a data set that is already publicly available. And when you request analysis of your own data, you can actually tell us what you want done to it. And these are the forms that you can get there. So we've been going quite a while now at various different data types. And here's a very quick overview of what we have in MGnify. Now, at the center on the right-hand side is actually a biome tree. Starting at the top in this yellow is engineered samples, soil, then aquatic environments, plant-associated, animal-associated, and human. In the innermost ring, it's just the number of studies. Then coming out is the number of amplicon, and then further out from that is metagenomics. And so you can probably see there's quite a lot of data there. So we have over 350,000 amplicon data sets, 34,000 metagenomes. So that's where they then unassembled, and over 31,000 assemblies. What's not shown on this particular graphic is the fact that we have thousands of metagenomes, assembled genomes, either available for the human gut plus other data sets that are going to be released imminently. So one of the things that we focus on a lot is having formal descriptions for our analysis pipelines. So we use the common workflow language. This is very geeky and very technical, so I'm not going to go into it. But this gives us the opportunity of two things. One is the portability of pipelines, and the other thing is to rapidly take tools and swap them out so we can evaluate new tools as well. And so we can very quickly understand the impact of a new tool within the pipeline and actually produce pipelines quite quickly. This has allowed us to do assembly. So we do both short read assembly, long read assembly, and hybrid assembly. We have a whole range of different analyses that we can run depending on the input data type as well. So really, this is a shout out back to Alex's work from the research, and just really to point out that, you know, we're operating at scales that are quite remarkable. So he talked about the human gut catalogue earlier. So we have, you know, over 210,000 genomes that we have in the MAG collection. This is causing some headaches on the informatics, but it's also very interesting. So we already have 170 million unique proteins in the human gut. If we put that with all our other biomes, we have over 1 billion protein sequences. And this is actually driving different areas of informatics. For example, AlphaFold 2 for the protein folding side, that used the magnified data for a lot of the underlying covariance models that it's used. So this is driving science in ways I'd never anticipated. So this service provides access to those genomes as much as possible. We make the species representatives available via the website for browsing, and the rest are available for download. So really, this is a shout out that we have lots and lots of data ready there to be consumed. We have programmatic access to all the different parts of MGnify, and would welcome interest and collaborations on the analysis of microbiome data. And that just leads me to thank my team and Alex for earlier presenting for the research site. Thank you.